Thank you very much. So my name is Vijay Rajagopal. I'm at the University of Melbourne in the Department of Biomedical Engineering. So today I want to talk about some excellent work that our PhD student Afshin Khadangi has done in the deep learning space to benchmark electron microscopy image segmentation algorithms. Before I begin that, just to give you a bit of a context of what it is that we do. So my group is called the Cell Structure and Mechanobiology group, and we work on a number of different projects, but broadly, what we're interested in is understanding how cells get their shape and architecture, and how does that architecture regulate cell function, and how might that architecture also be how that form may also be regulated by function itself. A particular interest to us is to understand how cardiac cells that make up a beating heart, how their architecture changes in disease, and we're interested in understanding, using computational models, the biophysical mechanisms that are involved in regulating that cell architecture, and again, how that architecture regulates cell function. So here's an image on the right here. We have these confocal images, for example, of the T tubules and cardiomyocytes under healthy conditions. And they change into, their architecture is the striated appearance that they have in healthy conditions is disrupted in disease conditions like heart failure, which is shown on the right here. Another example where cell architecture is very important and it's probably more dynamic is cell migration. And that's also an area that we've been slowly building up on, but I won't get into that today. Underlying all of this work that we do in understanding structure-function relationships in cells is that we couple experimental data with new computational models, so that are based on biophysical principles to develop our mechanistic understanding of that structure-function relationship. So let me just go through a couple of examples of how we go about doing this. One example is a relatively simple example to begin with is looking at red blood cells. So we've been working with a red blood cell and malaria specialist here at University of Melbourne, and they're interested in understanding how red blood cells flow through your blood vasculature, and how their biophysical properties change in health and disease. Particularly when someone is infected with malaria parasite, red blood cells can undergo changes to their shape, but they can also undergo changes to their stiffness as well. And one of the sort of questions that came up is, when both the shape and the stiffness change at the same time, does the stiffness really matter, or is the shape that matters more? If we can understand which one has a bigger impact on how the cell goes through narrow channels, we could through narrow channels like capillaries, we could then target treatments. In relevant disease conditions, we could develop treatments that target that one of those biophysical factors rather than the other. So what we did was we developed a microfluidic device where we put these cells, red blood cells, through narrow channels that were, incrementally, basically had a wedge shape, and they would basically get stuck wherever they got stuck is the furthest they could go, or what we call the minimum equivalent diameter that they could reach by squeezing through the channel. We also then built a computer model which involves parameters for the stiffness of the cell and the shape of the cell, obviously. So now what we could do with the experimental data of different of red blood cells going through narrow channels and under different conditions, and we had our biomechanical computational model, and we could then understand which one's more important, surface area or stiffness. So I'm going through very quickly here just to give you an overview. One thing we did experimentally is to change the stiffness of the cell using glutaraldehyde treatment, and we found that the minimum equivalent diameter, that position that the cell goes to, doesn't really get affected by changing the stiffness, and our model shows the same, as shown by this graph here. We also then changed the surface area to volume ratio using a treatment that changed the pH of the cell culture, and what we found is that you have a drastic change in the ability of the cells to go through narrow channels when you change that surface area to volume ratio. So this is an example of how we use experiments with computational models to develop mechanistic insights on what we're seeing in the experimental data to extract biological insights from mechanistic and biological insights from them. Going one step further as an example, and now getting into more of the reason why we did this EM stellar study. We're also, like I mentioned before, we're interested in looking at how cells get their, cardiac cells get their architecture and how it's regulated or dysregulated in disease. One of the things that we've been working on is studying how cardiac cell architecture changes in diabetes-induced cardiomyopathy. Here, what you're seeing is two images, cross-section. These are transmission electron microscope images of cardiac cells in control and a animal model of diabetic cardiomyopathy. If you think about a heart cell, to orient you on what you're looking at in this image, if you think about a heart cell like a cylinder, you're looking at the circular face of that cylinder, and this within that circular face, all these green contoured regions are your mitochondria, and the rest are, it's really packed with mitochondria, and then the rest are mostly myofibrils in there, which are the contracting proteins within the cardiomyocyte. If you look over the diabetic animal model, what we find is this smaller mitochondria, more of them, and they're almost crystalline architecture is a little bit more dysregulated. And so one of the questions we had was, well, how does the positioning of mitochondria affect the energetics in the cell? Because produced by mitochondria needs to be delivered to the myofibrils in a timely fashion so that the heart can beat effectively. So what we did was to develop a computational model that included a model of the biochemical reactions involved in the electron transport chain. We basically represent the electron transport chain reactions as a series of ordinary differential equations, and then we have a computational technique called the finite element method, with which what we can do is we can take images of the electron microscope images of the cell, segment them into mitochondria, myofibrils, cell membrane, so on and so forth, and then we can build up a computational spatial model of the cardiomyocyte. So we bring together the electron transport chain model with this geometric mesh using the finite element method, and then we can simulate the diffusion and movement of metabolites like ATP, inorganic phosphate, so on and so forth. And we take great care when developing these models to make sure that we're looking at experimental data with which we can validate model predictions for certain things that we can measure, and then that gives us confidence in our predictions and what the model tells us afterwards. So here on the left, we're showing you that our model, which is in these blue circles, follows the trends that people have measured experimentally for the phosphocreatine to ATP ratio as you change the oxygen consumption rate. Also, ADP concentrations are also the sort of cell-wide concentrations are changing in the same way that the experimental measurements have been done before. We've also measured, or I should say, simulated oxygen concentration through the cross-section of the cell, and which is represented by the blue curve here. And the experimental measurements sort of fall within that, or I should say, our model falls within the experimental measurements of that oxygen concentration as well. So we get confident in what the model predicts by comparing model predictions with the experimental data that's out there. And then what we can do is simulate different conditions. So here you've got the electron microscope image that I've shown you of the control myocyte, and then we overlay ADP to ATP ratio, ATP consumption rate, and there are many other variables in the model that I don't have time to show here. And then we also look at what happens when you have that function simulated, or the energetic simulated, on a diabetic cardiomyocyte. And you can see within this color map, it's in the control condition, it's fairly homogeneous over the cross section, but it's much more heterogeneous in the disease condition. This image is actually a frequency plot of the distribution of the ATP hydrolysis rate across that cross section. So the ATP hydrolysis rate, for those who are not familiar with bioenergetics, really gives you a measure of how quickly ATP is being consumed for contraction. And what you find is in the control myocyte, most of there's a sort of a narrow range of ATP hydrolysis rate, reflecting the almost uniform contraction of the cardiomyocyte. But in the disease condition, it's a much broader ATP hydrolysis rate across the cross section, reflecting the inhomogeneity that you see in the ADP to ATP ratio, and therefore inhomogeneity in force production in the cell as well. So that's sort of having shown you what we can do with these models, and particularly when we use them in conjunction with electron microscopy. The last thing I'll also mention is that we've been in this area of wanting to build models from electron microscopy data for quite a while. We've also developed methods where we integrated electron tomography images of cardiomyocytes with confocal images of ion channel distributions like Ryandian receptors. What we did was basically use in order to fuse information from two different image stacks. This is work from a while back now, before we got into deep learning. What we did was we used to extract the spatial distribution of ion channels from confocal images, and then we would extract the statistical distribution of those proteins relative to other organelles in the cell, and then we would simulate, using Monte Carlo simulations, we would simulate the position of those ion channels on electron microscopy data. And so we could develop more integrated 3D models of the cardiomyocyte. What we can do now is we can take these models and we can simulate not only energetics, but we also simulate calcium dynamics and mechanics as well, so that we can really go to town with, not just looking at images, but also gaining biophysical insight from those images as well, using our techniques. So so through the years of developing these models using electron microscopy data, one thing we've always hit as a bottleneck is segmentation of these images. Up until Afshin came on board, a lot of the work we did was basically using Fiji or ImageJ and manual IMOD and manual segmentation algorithms, or I should say manual segmentation, to extract the images and generate meshes from our models for our models. But since Afshin came on board, we've been investigating, all these different deep learning algorithms that are out there. But what we found was that every new algorithm out there was presenting a positive result, that that their algorithm was really working to segment particular structures or particular application areas. So what we wanted to do was we really wanted to know, is there one method out there that we should be using, one state-of-the-art network that we should be using, or what kind of comparisons have been made already? And we found that there's really not much that's been done out there for benchmarking these algorithms, and to give people an objective view of how good their algorithms are. So what Afshin's done is develop this Google Colab notebook called EM Stellar, which allows you to compare the outputs for a given data set. You can compare the performance of a variety of different deep learning algorithms, which I'll just go over for a few minutes now, and then Afshin will give you a little demo of what it looks like. So this is the overview image. Basically, we've developed this Google Colab notebook involving several state-of-the-art deep learning architectures. So UNet, SegNet, CDeep3M, ResNet, and VGG. EMNet's actually one of our own deep learning algorithms as well. We've published a paper recently on EMNet, and it's sort of a combination of Prelude and ReLU, or I should say an advance from Prelude and ReLU deep neural networks. But you can find out more details about that in our paper, or certainly talk to us after this presentation. So these algorithms basically have been implemented in this Google Colab, and so a user can come in with their raw data and raw data and their manual segmentations of a sample of the data set, and basically train these algorithms. Train these algorithms on this data, and we also have once they give you outputs, we've also then shown. Whoops, sorry, I've just lost my screen. Here we go. We've also then provided a number of different metrics so that you can look at how the algorithms perform across a wide range of error metrics to give you a sense of how consistent the accuracy of the algorithm is for your particular data set. And what I'm going to show you is some of the things that we've learned from using this Google Colab on a couple of data sets that we've used. The data sets that we've used are in developing this benchmarking tool are two different data sets. One is a FIB-SEM, so it's a focused ion beam scanning electron microscope image stack of cardiomyocyte ultrastructure. And another one is a neuronal image stack that's previously been used for EM segmentation challenges as well. Just to give you a view of what the images look like, this is one of the images from the cardiac data set, and this is one of the images from the neuronal data set. And we basically trained the algorithm the algorithms that are on that Google Colab to segment the mitochondria within these two data sets. The cardiac data set's pretty small. It's only got like 30. The entire data set is much smaller than the neuronal data set. But basically, to be very basically, we took subsamples from this, patches from these data sets, passed some of them for training, saved some and then saved some for validation and validation purposes. So and then looked at the performance of all the algorithms in the validation phase. Here's an output that you can of a heat map that you will get from using Google, our EM Stellar Google Colab. What you see on the bottom are all the different algorithms that we've implemented in the platform, and the vertical sort of y-axis represents all the different error metrics that we've calculated for each of the data sets. So on the left here is the neuronal data set, on the right is the cardiac data set. And one of the things we were looking for is there one algorithm that seems to work for all metrics? And you can see that it's very hard to find any one algorithm that fits all of them. As in, it's not, VGG may look like it has a relatively great F1 score, but it doesn't seem to perform that well if you look at the sensitivity, for example, in that heat map. What we did find, and this is shown in the black box here, is that EMNet's variants seem to be, seem to have reasonable robustness across these error metrics. But that's not to say that we think that that's going to be the same for every new data set that comes across. Indeed, what we did is also look at whether the performance that we're seeing, variations that we're seeing, is a reflection of the data that we're looking at. And you can see here, just broadly speaking, all the blue bars here represent the convergence times for segmenting cardiac ultrastructure, and then the pink ones are all the neuronal ones. And so you can right away see that looking at just the type of data you get affects the performance of the algorithms. But also, if you now looked at, say, comparing, for instance, let me see, the cardiac EMNet V12X, it looks like it performs, it has a smaller convergence time with the cardiac data when compared to the neuronal data. But if you then, if you compare it to the neuronal data, which is over here, you could then try to find another algorithm that performs. You may find that another algorithm performs worse than EMNet V12X in this instance, but there are other instances where it doesn't work so well. And so if you go through our paper, you'll find that we find quite a lot of variation in performance because of the structure of the data set. We went and also tried to look at whether the performance of the algorithms is dependent on the sort of number of the complexity of the algorithms themselves, i.e. the number of operations that you have to use and the size, the number of parameters in the model. And you can see, when it comes to the VRAN score, some algorithms perform much better, and it's not that they don't have that many parameters. Certainly, looking at this plot here, you get quite a good performance with relatively few parameters compared to other architectures that have been published previously. What we also then did was overlay, and you can do these overlays in your own in EM Stellar yourself for your own data set. We show how these different algorithms have different positives and advantages and disadvantages. And in this image, what you're seeing is that when you look at each of the segmentations of the raw data for each of the different algorithms, we find that in this case, CDeep3M produces less false negatives compared to the other algorithms, just by and so this is sort of the power of using this Jupyter Notebook. You get to see where you get an overview of where the algorithms perform well and where they don't perform well, and you can choose what you want to do with that information for your own purposes. So I'll finish off this before Afshin gives his demo by just saying that our recommendation from what we've done so far with EM Stellar is that if you have a relatively small data set, using VRAND and the VInfo or F1 score is quite a good reliable metric to get accurate segmentations. And you could do majority voting or average voting to sort of not have to decide on one particular algorithm. You can just use a bunch. You can choose the best or the majority voting approach to choosing the best segmentation. But you will, as you can see already, you will what you choose affects whether you get more false positives or false negatives. So with this, I'll thank you all for your time. And Afshin, did you want to share screen and show us your demo?
Hi everyone. Hi Vijay. Yes, absolutely. So I'm going to share my screen. Screen one. Okay. Thank you very much, Vijay. Since this takes a little bit to run, I will just submit the running, and the details can be found on our repository. So as Vijay mentioned, we have deployed the EM Stellar on Colab. This gives more hands-on and hassle-free access to the user to access the hardware and software requirements simultaneously in one place. However, one of the reviews of our paper raised a really good point and concern about data privacy, as Colab requires the data to be uploaded to the Google Drive. And sometimes, as you be aware of, the data that we are dealing with sometimes are large, so it takes some time to upload, to download. So what we did, we implemented a Python script which takes the directories for the local install Fiji and ground truth images and predicted results from different methods. So to benchmark, we have got VRAND and VInfo scores after thinning, and other non-standard metrics, standard metrics, including 11 other metrics as F1 score, accuracy, specificity, sensitivity, negative and positive predictive values, and more importantly, Jacob score. So the program is being run on the left side. So what happens? The user has got some ground truth images for the segmentation results, and these are the ground truth test masks that we are interested in evaluating the outputs of algorithms. So, for example, here we have got 13 different folders, which is actually representing the outputs from different network topologies and methods. So here we have got CDeep3M, we have got EMNet, we have got UNet, SegNet, and etc. So the user needs to just add the segmentation prediction masks from different algorithms to the designated directories as shown here. And then they also need to specify the ground truth masks in other designated folder. And what happens, the script goes through different directories and groups the files together. And once we have this temporary folder, the algorithm goes through these stacks of images. So we convert the slices into the stacks. And as you see on the left side, the algorithm goes through and iteratively calculates the standard metrics and non-standard ones, which are here VInfo and VRAND score. We were interested in VRAND and VInfo score, and we, on the paper, actually, we really emphasized and highlighted these two metrics because it gives a better perspective from the over and under segmentation perspective, which is pretty important in some domains, including the neuroscience, and also in our application, cardiomyocyte segmentations, because sometimes we are interested in segmenting mitochondria and myofibrils and Z-discs in one place, and over and under segmentation there is really, really important. And after the VRAND and VInfo are calculated, the algorithm goes through obtaining and pooling those standard values, standard metrics that I just mentioned, including F1 score, negative and positive predictive values. We also, for the implementation, we also ask the users to provide their local installed Fiji app, because we have integrated, we have used PyImageJ to integrate with Python and utilize some of the functionalities of the Fiji, which are a lot of metrics out there in Fiji, including VRAND and VInfo score for our applications. So after the script has been run and we have extracted the scores that we mentioned, so yeah, as you see here, so what happens? So what happens, we extract the segmentation metrics based on the VRAND and VInfo score, and then we retrieve the standard metrics. And what happens as a result when we run the script to this script actually gives two outputs to the user. So one is a data frame output as HTML file, which includes the a data table actually, which represents the scores and yeah, the scores and for different methods, as you can see here. So for example, we can see that the sensitivity is much better. CDeep3M performs really well compared to the other methods in terms of sensitivity, but for the specificity and positive predictive value, it is even worse. So,
if you could, sorry to interrupt, if you could reshare the screen, at least on my side, I don't see your screen since the mute topic happened a moment ago. So if you could stop sharing and share again, maybe we'll come back on. Thank you.
Yeah.
Yeah, we see it now.
Yeah, sorry. So yeah, so one of the outputs is the data table HTML file, so the user can go. So what happens in Colab, these are on the notebook, but what happens when you run the script, we were interested in giving the user two different outputs, which was one data table representing different scores, and the other one, I hope that this time you can see also my screen. So this one is the heat map that Vijay also has shown to you during the presentation. So we give the user also a heat map of different.
Afshin.
Oops.
We're not able to see. I'm not able to see at the moment.
Yeah, I see a grid of numbers with yeah, it looks like a heat map.
Oh no,
okay. This is a heat map. Yeah.
Yeah. Yeah, so we show the users the heat map of different methodologies and yeah, the corresponding scores. So this is actually much about the implementation of the MSTELLAR on your local machine. And yeah, we have also provided the users the Google Colab access, so they can. We have also provided the instructions, so the users can go to the Google Colab and mount their Google Drive and upload their data and perform exactly the same analysis that we just showed to you here. So, any questions?
Thanks, Afshin.
Yeah, that's great. And the flexibility of having both the local deployment that you just demonstrated and a Google Colab is really neat because yeah, if you don't want to upload everything, it's really essential. So I just want to thank you. It's not really a question. I do have a question, but first of all, this is a mammoth amount of work to benchmark so many networks. And just one curiosity, did you have to re-implement all of them, or you found a way to port implementations that are published into a general location?
So what we did here, so from some of these networks haven't been utilized in biological image segmentation domain. For example, UNet is much popular because it gives you a better generalization capacity. It has got a good number of the parameters, which is not that much as compared to the other networks architectures. But yeah, it has shown much robustness. But what we did, we were interested in implementing UNet-like architectures where we used, so for example, UNet comes with one feature extractor in contractile path and one expansive path as decoder and encoder. So what we were interested, we were interested in encoding the data into features, abstract features using different network topologies. Yes, so we used the previously developed algorithms on the biological domain, and we were interested in using other network topologies like ResNet as the encoder and integrating in the UNet-shaped architecture.
Okay. So another thing I'm curious about is whether you've considered to do this for fluorescence microscopy data, where some of the people attending today are interested in EM for sure, and others are interested in light microscopy. So I was wondering how far away would it be to just deploy exactly the same infrastructure for light microscopy?
Yes. So from our experience, we had the opportunity to implement different deep neural networks and even elastic. So we have also compared elastic and Weka in our implementations and in our paper. And each of these methods provide their unique way of advantages. But from our perspective, from our experience, implementing and scaling up these methods to other major modalities, including light sheet microscopy, as you mentioned, is just like implementing the EM in the EM domain and EM images. So I believe it will be the same procedure. But yeah, it's definitely one of the future studies that we might be interested in